In this video, we will cover transmembrane transporters and also action potentials. So these are two huge concepts within cell biology and within other biology classes. So it's very important that you watch this video. So the main thing that we have to focus on right now is the difference between transporters and channels. So what is a transporter? A transporter will directly bind to a specific solute and it will transport it. So let's say that we have glucose, right? So let's say that we have glucose over here, and I'm not going to draw out glucose. So this is glucose, and we are going to write it as G, and glucose is going to bind to the active site, right? Well, not to the active site, but it will bind to the pocket of this transporter. And here you see glucose is going to be transported into the extracellular side, right? So let's say that this is the extracellular side, right? This is the environment. And this is going to be the inner cell, right? Okay, so we want glucose to get out of the cell in this example. And so what happens here is that we are going to induce a conformational change. So this is a conformational change. We are physically moving the transporter. It is shifting its shape. Okay? So right here, it's pretty specific. Okay, so this is very specific. Very specific. So for instance, if I had something like, let's say, an amino acid. Let's say that we are trying to transport an amino acid. Just an example. If the amino acid tried to get into this transporter when it's empty, it's not going to do it. It's not going to be able to enter the transporter because, again, this transporter is very specific to glucose. So, for example, let's say that whenever it changes shape, it molds itself into the shape of glucose, and glucose can fit right into that shape like a key to a lock. So because of this very specific conformational change, we can only transport glucose. Amino acids, ions, whatever, it cannot pass. So transporters are very specific. They induce a conformational change, and they take something from the from either the external or internal, and they shift it into the other side. So for instance, I can have solutes like glucose go in here and then come out through the other side. And I can have something coming from the inside going on to the outside. So that's why we have these little two arrows, right? So we can go something from inside to outside, or we can go from outside to inside. It really doesn't matter. So that was a transporter. Now we'll talk about the channels, right? So the channels are a little bit different. So channels, they rely on the pore size and the electric charge of whatever is transferring. So really, you take into consideration the charges for channels, which is pretty easy because we have C for channel, we have also C for charge, right? And right here, they don't really change shape, okay? This is more like a gate, okay? It's more like a gate. And they also have pore size. Let's write that down. So we have pore size. We have different sized channels, right? And really, because since we're charged and we want little things to pass through us, usually little things, you'll often see water being transported, and you'll often see ions being transported, right? So these are going to be transported at the same time. Now you're probably wondering, well, hey, if we have two different things able to go into the channel, does that mean that we're specific? No, actually. So channels, they're not specific. So we have not specific. So let's write that, specific. So what do I mean by that? Let's say that we have ion X, right? Ion X. And let's say that we have ion Y. So notice that ion X and ion Y, they both have positive charges. That means that they have the same charge. And let's say that they have the equal amount of charge. You're probably wondering, why did I write this Y, or this ion Y, to be small? Well, that's just to show the size difference. So let's say that ion X is able to go to the channel, right? And we're going to go to this side right here. Okay? Well, let's make this into a better one. Let's say that ion X is going to be on this side, and let's say that ion Y is going to be on this side as well, right? Because of pictures, you know. Anyways, so let's say that ion X can enter this channel pretty easily, and it could go into the inner cell. Now, because ion Y is smaller than ion X, it can also go into the cell or into the channel. So because I'm smaller, I can go inside as well. But what if, and let's do this, what if we have ion B? And what if it's big, right? Would it be able to fit into the channel? Most likely not. Most likely it'll try to go, but because it's bigger than ion X, it will not be able to go into the channel. And so really a huge factor in determining whether ions can cross in channels is that the size matters, okay? And also the charges. So if I have a small ion like this right here, and I have a negative, maybe I'm not able to go into the channel. Maybe this is really specific for positive charges. Maybe one channel is specific for negative charges, right? So depending on the channel type, you'll have a kind of like a selection between ions and their charges. So to recap, channels compared to transporters, they are not specific. However, they do have a pore size and a charge selection. So for instance, if I only want positive charges or positive ions, then positive ions will flow through. If I only want small ions, then only small ions will pass through. Now we have different channels. So channels could change depending on where you're looking at in the body, right? We could be looking at the brain, we could be looking at the kidneys, right? So depending on where you're at, the sizes could change and the ion charges could change. Okay? So yeah, so these are like the main differences between transporters and channels. And here we have a channel in action. So this is an aquaporin, and in the last video I defined what an aquaporin was. Essentially, it could be beta barrels within the membrane. But what happens here is that water is able to go in and out of these aquaporins, and they can actually help us regulate the volume of a cell or just nutrients, right? So again, nutrients, ions, water, they can go through the aquaporins. And here we have a beautiful picture, right, of this in action. Okay, so what kind of transport does this use? Right. So this actually uses a passive transport. And what passive transport means is that it is simple diffusion. It is simple diffusion. Right. So right here, simple diffusion does not use ATP. So it does not use ATP. So no ATP. Right? It just uses the concentration on either side. So let's say that we have more waters on this side, and we only have a little bit of water on this side. So because there are more waters on this side, we can say that the concentration gradient is going to push the water into the inner cell, right? So this is the environment, and this is the inner cell, right? Eventually, it will reach an equilibrium. So eventually, here we have like seven waters, and over here I have 3. Eventually, it will reach equilibrium, and I will have 7 as well, right? So we're going to have an equal amount of shared water, and that is just simple diffusion, right? So in that example, you have to imagine that there's more water than what I drew, okay? Okay. So again, these are integral membrane proteins, and they're just there to enhance water flow. So this is enhance water flow. And sometimes you'll see on an exam, they'll say, oh, we're going to add aquaporins to do what? And then an answer choice will be to affect the direction of water, or to increase the amount of water that goes into a cell, or actively transport water to go into a cell. That's going to be wrong. You're not going to do active transportation because there's no ATP being used. Okay, we are depending on the concentration gradient. Okay, so this itself, by itself, it doesn't affect the direction of transport. It just establishes equilibrium. Okay, so establishes equilibrium. Right there. So you see, sometimes water will diffuse through the transmembrane, but most of the time the water will go through this channel and it'll have a great time. Notice that this is not a transporter because we are not making a conformational change. The channel is always open. Okay, that's why it's always in equilibrium. This does not change the shape. However, sometimes within organisms, we don't want to have so much water in our cells. So we have to actively transport water out of the cells, right? So how do we do that? Well, within animal cells, we can use the sodium-potassium pump. So we can use the ATP Atpase pump, right? And some people call it that, so they call it like the sodium-potassium pump. Some people call it ATP Atpase pump, and it's okay, right? And the main thing that you have to remember about the sodium-potassium pump is that we have a mnemonic, and it says 3, 2, 1, Nokia. So what is 3, 2, 1 Nokia? So 3, 2, 1 Nokia stands for 3 sodium out, right? And then we also have 2 potassium in per 1 ATP. Okay? So that's 3, 2, 1 Nokia. And trust me, that mnemonic has saved my life so many times. I can't tell you how many times I've been at the mall and people just push me against a wall and they're like, how many sodiums are transported out of the cell in a sodium-potassium pump? And I say 3 out. Okay, that's just, it's going to save your life. Promise. Now, within the plant cells, we have a different thing. So right here, notice that we have a cell wall. So the cell wall will actually maintain the shape of the cell. So that kind of protects us from exploding, right? So from cell lysis, it doesn't really happen for plant cells, okay? Because we have a cell wall. It does happen, but not to a great extent, right? And we also have kind of like the proteazoan, right? So the proteazoan is going to have a special water collecting vacuole. So again, a vacuole is kind of like a, it's like an organelle that just keeps control of the water, the ions, the nutrients. And what this vacuole can do is that it can take the water, right here, it can take the water and it can shoot it out. It can shoot it out into the environment. And so protozoans, like bacteria, they can take water from within their cells and they can forcefully shoot it out against the environment. And that's pretty interesting because most people don't know that. They don't know that. And I knew that. That's why I'm teaching it. So yeah, these are pretty cool. So again, the most relevant for most classes would have to be the animal cell. Protozoans are not really talked about, but animal cells are always talked about. And now we will be talking about the tonicity of a environment. So tonicity is the relationship between water and the solute within a system. So we can have 3 different tonicities. We can have isotonic, hypotonic, and hypertonic. Isotonic means that the amount of water and solute is equal. So we have equal water and solute. So we're just going to call solute S. Okay. Now hypotonic means low. Okay. So hypotonic means that we have a low amount of solute within the environment. So we have low solute. in environment. And then we have hypertonic solution. So hypertonic means that we have high solute in environment. So now sometimes students, they have a difficult time saying the difference. You know, they don't really see the difference, or they don't see how hypotonic and hypertonic can do the effects, right? So for instance, isotonic is pretty easy to see that water goes into the cell and it also leaves the cell because we have an equal environment of solutes. That's pretty easy to see. But hypotonic and hypertonic are a bit more difficult. So what I personally see is that for hypotonic solutions, we have a low solute in the environment. So my solute, let's say, is going to be pink. So we have little pink dots, okay? And we have little pink dots. Now, compared between the cell and the environment has a little bit of solute, but the cell typically has more solute. So there's a difference. There's a difference between the cell and the difference between the environment. Again, the environment has a little bit of solute in hypotonic solutions, but the cell, the internal environment of the cell, has more solutes. So this is not a good color for a blood cell. Let's use green. So this one is going to have more solutes. Right, we have just a bunch right there. Okay? But notice, here's the difference, and here's why you need to remember this. Right here, we have more water outside, right? But because the inside of the cell has so much solute inside the cell, there is a little bit of water. So there's not too much water in here. Just a little bit, okay? Just a little bit. Because there is more water outside than inside the cell, the water from the outside wants to go into the cell. Now I'll recap. In a hypotonic solution, there is a little bit of solute outside of the cell. Because there is a little bit of solute outside of the cell, there is more water. Now, inside of the cell, there is a lot of solute. Because there is a lot of solute inside the cell, there is a little bit of water because the solute takes up a lot of the space in the cell. Because there is more water outside of the cell and a little bit of water inside the cell, the water from the outside of the cell wants to go into the cell, and this causes cell lysis. So it makes the cell explode. Now, in a hypertonic solution, there is a lot of solutes, right? So there is a lot of solutes, like just too many solutes, okay? Now, inside the cell, there is a little bit of solutes. Okay, so inside the cell, there is a little bit of solute. But inside the cell, there is a lot of water. So the water is going to be orange. Okay? Because if there is a little bit of solute inside the cell, then there has to be something else. And that something else is water. So there is a lot of water inside the cell. Now, because in the external environment, there is a lot of solute, there is a little bit of water. Because the solute takes up space in the external environment. So because there is a little bit of water outside, and there is a lot of water inside the cell, the water inside the cell wants to go out. So we always go from high water to low water. So now the water is going to go and is going to go into the environment. So let's recap. In a hypertonic solution, there is a lot of solute outside of the cell. Because there is more solute outside of the cell, there is a little bit of water outside of the cell because the solute takes up space. Now, in the cell, there is a little bit of solute. Now, because there is a little bit of solute, we have to have more water to take up the space. Because there is more water inside the cell in a hypertonic solution, the water goes from high to low, meaning that the water will exit the cell and go into the environment. So there are two ways to see it. For a hypotonic solution, you can see that the solute goes out of the cell and the water goes into the cell. And for a hypertonic solution, the solutes go into the cell and the water goes out of the cell. So some people like to see the opposite, and some people like to look at it in the viewpoint of water. That's how I like to look at it. But whichever you like, feel free to use. Now, within hypotonic solutions, plants, they're going to absorb water, so they don't explode. Why don't they explode? Well, because they have the cell wall. So the cell wall prevents cell lysis in this case. So turgid just means rigid, and this is kind of like the ideal position for plants. But right here, whenever we have some shriveledness, plant cells will actually plasmolyze. So plasmolyze just means to shrivel. So it's very interesting. Sometimes you'll see that vocabulary within exams. Just plasmolyze means to shrivel. Turgid means to be rigid, means to be swollen up. And flaccid just kind of means neutral. Right, so within every eukaryotic cell, and also plant cells, we have specific types of transporters. So in one cell, we may have a nucleotide transporter, a sugar transporter, an amino acid transporter, and the sodium-potassium pump transporter. And that allows us to have multiple things coming in and out of the cell. And again, transporters are very specific. So the nucleotide transporter is only going to transfer nucleotides, and the sugar transporter is only going to transfer sugars. So a nucleotide cannot enter this transporter. A nucleotide has to enter this transporter. And again, most transporters transport organic, which means it's made of carbon, and it has hydrophilic components. So transporters transport organic, hydrophilic items, right? So they're water soluble, they're small, and they're made of carbon. So this is a passive transporter. And a passive transporter does not use ATP. So no ATP used. And what happens here is that a very specific transporter is going to transport an item with the concentration gradient. Okay, so let's say that over here on the outside of the cell, so this is the environment, let's call that enviro, there is a lot of glucose, and we need to get that glucose inside the cell. Because there is more glucose outside of the cell, we're going to use this concentration gradient and we're going to push the glucose into the cell. Now we go from high concentration, so we go from high and we go into the low concentration. So from high to low, that's okay, that's good. You know, we're going to be using just this gradient, so there is no ATP used. And of course, when a transporter is operating, it's going to undergo transformational changes, right? So this is a conformational change. This is a conformational change. And you can see the mechanism where the glucose goes in here. Here's a little glucose. It's in here like a gate, and this gate is going to open, and this glucose will exit the transporter. And this is active transport. Now, some people, they call these pumps. So this is always done by a class of transporters called pumps. So what I'm saying is this transporter, they're called pumps. And you can think of it because whenever you're pumping something, you're doing energy to pump that water or items, right? So let's say that I have a flood in my house and I need to pump the water out of the building. Well, I have to physically pump the machine to pump the water out, and so this requires energy. So this is ATP driven. And right here, this is the extracellular space. We're going to call this enviro. And this is going to be called the inner. So notice that in the environment, there is more items outside. This could be ions, this could be anything else, right? And in the inside, there's a little bit of that item. But notice that this item right here is going to be driven outside. And you're probably wondering, well, hey, we're going from a low concentration to a high concentration. So this is a low concentration, and this is going to be a high concentration. And you're probably wondering, well, we can't go from low to high. Well, not naturally. We can go from low to high if we input energy. Okay, so the only way that I can physically push an item from low concentration into an environment of high concentration is to couple it with ATP or any other energy source. And so in most cells, especially eukaryotic cells, the energy that you use is ATP. But sometimes, such as bacteria, you're going to be using light. So a good example of this would be the Bacterial Rhodopsin. So this was Bacteria Rhodopsin. You can actually see the 7 membranes right there. So 1, 2, 3, 4, 5, 6, 7. That is a 7-membrane Bacterial Rhodopsin. And what it does is that it uses light to generate protons. So over here you would have a high concentration of protons, and we would be using light to pump protons into the extracellular space. And over here is a sodium-potassium pump, most likely. And here we have a coupled pump. So sometimes, let's say, sodium would enter the cell because sodium is high over here, and glucose would then exit the cell, because when this transporter opens, sodium enters the cell from a high to low concentration, and then here glucose, which is very small, would go from a low to high concentration. And so to recap, sometimes you have a coupled pump where the outside space has a high concentration of something, and the inside space has a low concentration of something. So we go from high to low, and that is favorable. That doesn't need energy. But when this opens, the other item, such as glucose in this example, goes from a low concentration into a high concentration. It kind of piggybacks off of this gate opening. So this right here is always unidirectional. So active transporters are always unidirectional. Meaning that one item always goes in one direction. So when sodium enters the cell, sodium is not going to go back out of the cell. No. When glucose goes out of the cell, it's only going to go in one direction. It's not going to go back. Okay? And speaking of directions, we have some other pumps that can do other directions. And now we will be talking about transporters and how they're defined as Uniport, Symport, or Antiport. Now, the main thing that you have to remember with Uniports is that these are not pumps. These are not pumps. So right here, we can't go from a low concentration into a high concentration. That is not possible. Over here, you can only go from high into low. And that's kind of like a rule for Uniport, and that's very special for Uniport. Now, within these transporters, we're going to be exploiting the gradient. So we're going to assume that outside we have a high concentration, and inside we have a low concentration. So we're going to be using this high concentration to exploit movement. Now, Uniports, they only transport one item. So this is just one item. And it goes in one direction. So this is one direction. Okay? So we go from high to low. And this is just one direction. So it's not possible for this item to go this way. No, it has to go in the specified direction, and that's usually into the cell. Or if this was a high concentration and this was a low concentration, then the item would go out of the cell. But in this example, we go from high concentration outside to low concentration inside. Now, for Symport and Antiport, these are called coupled transporters. And they're called coupled transporters because they move two things at one time. So let's call this X. Let's call this X. And let's call this Y. Now, when X goes from the high concentration into the low concentration, it allows this transporter to open. And when this transporter opens, item Y can also enter the cell. So this is kind of like the piggyback. This is the piggyback method. And a good example of this, which we'll talk about later, would have to be the sodium and glucose co-transporter. Now, for here, we have the antiport. Now, the antiport is very similar to the Symport, except we have opposite directions. So for the antiport, we have opposite directions. Let's go opposite directions. So right here, we can see that item X and item Y. Item X goes into the cell, but item Y goes out of the cell. So that is the antiport. So the Symport is the same, S is for same, and A is for antagonist, which means opposite in this case. So again, we use the high concentration of X to go into the cell, and when this gate opens, this Y can go out of the cell. So here is the glucose sodium transport, and notice that we have a high concentration of sodium outside of the cell, and we have a low concentration of glucose out of the cell. So we have a low concentration of glucose outside, but we have a high concentration of sodium outside. Right? Inside the cell, we have a low concentration of sodium, and inside the cell, we have a high concentration of glucose. So over here, this transporter is going to open, and when it opens, sodium, which is a high concentration outside, goes into this pocket. Now, when it enters this pocket, glucose, which is a low concentration outside, also enters the pocket. So here we have both the sodium and the glucose inside the transporter. Now, when this gate opens, sodium is able to enter the cell, and also glucose. So this is called a Symport. This is a Symport. Because notice, both sodium and glucose went into the cell. And also notice that we pushed glucose from a low concentration into a high concentration. So we kind of exploited this method. This is like the piggyback method. So when sodium is able to enter, it opens its gate temporarily. Now glucose can sneak inside like a spy, and when it does, this transporter can open and sodium and glucose can both enter the cell. So we push glucose from a low concentration into a high concentration, and that is very important for us when we have to metabolize sugars for energy. So without this pump, most likely you would have a hard time surviving in the real world. Within our cells, we have this mechanism that allows us to do this indefinitely. So sodium will always go into the cell and glucose will always go into the cell. But how do we do that? Well, over here we have an active transporter. Okay, so it's an active transporter. And what I mean by active transporter is that we are pushing glucose from a low concentration, so low gluc, into a high concentration. So we have high glucose. So sodium goes into the cell, and glucose also goes into the cell. And so this is a Symport. This is a Symport right there, right? Because remember that over here we have a high concentration of sodium. So since we have a high concentration of sodium, it wants to go from a high to a low. Now glucose is inside the cell and is being metabolized, right? So it's being oxidized and it undergoes reactions. But what if we need to get rid of the glucose? Well, we have a high concentration of glucose over here inside the cell, and so I don't need to use any co-transporters. I could just get out, since I go from a high to low, I can use a Uniport. So again, we have a Uniport right here. And a Uniport is just going to take one item at one time. In this case, we're only going to be taking glucose. So we make glucose go from a high glucose into a low glucose, which is good. Now, you have a buildup of sodium inside the cell. So how do I get rid of this excess of sodium? Well, I can use the sodium ATP pump. That means that I can get rid of 3 sodiums right there, and I can pump 2 potassiums into the cell. Now, because I'm getting rid of sodiums, I'm going to have a decreased concentration of sodiums inside the cell. Because there is a decreased level of sodium inside the cell because of the sodium-potassium pump, the sodium outside is going to be relatively high compared to the inside of the cell. Because of the high concentration of sodium outside of the cell, I'm going to be driven into the cell to undergo equilibrium. And when that happens, glucose is going to go into the cell as well. So you can see that the sodium-potassium pump regulates the intake of glucose. So we can say that the sodium-potassium pump maintains glucose uptake. So it gets rid of the excess amount of sodium into this of the cell, and because there is a low concentration of sodium in the cell, the sodium outside can go into the cell. So very cool. Now, within plants, fungi, and bacteria, so we have plants, fungi, and bacteria, they use hydrogen pumps. So they use hydrogen pumps. Okay? And they use hydrogen pumps to create an electrochemical gradient. Because hydrogen has a charge, a positive charge, it's going to create an electrogradient. And this is going to be used to create energy for the cell. So it can do Krebs cycle, it can do the Calvin cycle, it can use photosystems and whatnot, and it uses this hydrogen pump. Now, animal cells also use hydrogen pumps. So sometimes you have a lysosome, which is very acidic, it has a low pH, and the only way that you can put so many hydrogens in one area would have to be to use ATP. And so really, hydrogen pumps, for the most part, they're going to be ATP driven. So this is ATP driven. Now, notice over here we have a co-transporter, and this is a Symport. Notice that we have a high concentration of hydrogen right there, and we go from a high to low. And when this opens up, the solute also enters the cell. And so we can have Symport, so we can have pumps, mainly pumps for plants. Now, the hydrogen ATPase pumps, they can be found in membranes of organelles. So for instance, you can find H pumps in organelles such as the lysosome, and also the vacuole. And that makes sense because sometimes you have waste that is from the plant and it goes into the vacuole, and you need a really acidic environment to break down the vacuole, or to break down the waste. And now we begin the portion of the lecture that focuses on membrane potentials. So membrane potentials, they are governed by a permeability of specific ions. So a membrane potential. And from now on, I'm going to be calling membrane potential MP. Okay? So the MP of a cell is really dictated by how many ions are in the extracellular side and the intracellular side. And the distribution of ions on either side, it will actually create a potential. So let's say that we have too many sodiums outside of the cell and a little bit of sodiums inside the cell. Now, because there is a difference in charge, there are more charges outside than there are inside, we can actually create a potential. So we can have a greater positive charge right here and a small positive charge there. Now, the system will try to equilibrate itself. It will try to go into equilibrium. And when this happens, we can have a potential. And so what you see here are these little curves and these little strips. And this curve is called an action potential. So this is called an action potential. And this action potential is the entire basis of movement. So whenever your muscles are exercising, or whenever you're walking, or when you're breathing, you have trillions of action potentials going on in your system, and that allows you to move. And an application of an action potential is actually the venous flytrap. So this is a venous flytrap, and you may know that a venous flytrap is a plant, but you're probably wondering, hey, I thought plants couldn't move. Why is this venous flytrap able to eat a fly? Well, most likely it's hungry. I mean, I would bet that it's hungry, that's why it's able to eat a fly. But actually, it uses action potentials to capture its prey. So whenever a fly touches a little hair right here, the action potential is triggered, and so we see an influx of sodium entering the cell. And when this happens, we actually depolarize the membrane. We'll talk about that later. And when the membrane is depolarized, it allows the fly to be captured by the venous flytrap. And so now we will watch a video of the venous flytrap in action. And this was created by the BBC, the British Broadcast Company.
The venous flytrap. Like the sundew, it makes itself very attractive, oozing nectar across the brim of each leaf. But any visiting insect had better watch out for these 6 tiny hairs. This fly has to tread carefully. If it strikes one hair, it can carry on feeding, but a timer has been set. A second strike in less than 20 seconds, and the fly is doomed. An electrical impulse is triggered, and the leaf snaps shut in just a fraction of a second. The tips lock together like prison bars. If the fly is very big or very small, it may just manage to escape. But most are trapped and die. Ten days later, the trap reopens. All that remains is a husk. The plant has finished its meal and resets itself for its next victim.
I hope that fly is okay. I mean, he looked like he was having a bad time, struggling. You know, if that were me, I would have escaped. I'm just built different. So this is an action potential. And here we have a mixture of sodium and potassium. And so this dance that sodium does with potassium will create an action potential. But before we can get into what an action potential is, I have to introduce an equation. Now you don't need to memorize this equation, it's just there to help you understand what's going on and why we have a negative number. Okay. So over here we have that the membrane potential is going to be equal to 61 millivolts multiplied by the log of, and let's just focus on one ion. Let's say it's the log of the potassium inside the cell. Let's say outside. Outside of the cell, divided by the concentration of the potassium ions inside the cell. Now within a cell, there is more potassium inside. So this number, let's say, is going to be a very large number. Let's say that it is the log of 10,000. And let's say, hypothetically, that there is only one potassium ion outside of the cell. Because this is a larger number, this will be a negative number. So what I'm saying is, when you're working with logs, the denominator, if the denominator is larger than the numerator, then we will have a negative number. Because this is a very, very small number. This is like 10 to the, let's say, 0.00001, right? And 10 to the 0.001 is some negative number. So what I'm saying is, because there is more potassium inside the cell than there is outside of the cell, this is a negative number. If the bottom of the log is larger than the top of the log, this is a negative number. So hypothetically, let's say that this is equal to negative 84. I don't know if it is. I don't really care. I just know that it has to be a very large negative number. So within the eukaryotic cells, typically we have a resting potential of about negative 20 millivolts all the way to negative 200 millivolts. And that's just resting. Nothing is happening and we don't have an action potential. But the reason why this is a negative number is because there is more potassium ions inside the cell than there is outside of the cell. So now, when sodium enters the cell, it has a positive charge, and this potassium also has a positive charge. When the sodium enters the cell, too much sodium enters, and the potassium is repelled by the positive charge, and so it wants to exit the cell. So if we just look at the potassium, just looking at it by itself, we would have, after the sodium enters, we would have more potassiums outside of the cell and a little bit of potassium inside the cell. So this would be a positive number. And that is why whenever an action potential occurs, it shoots up. It goes to like 30, well, it goes to about 30 millivolts in the positive direction. Because this number is bigger on the top, it has to be a positive number. When sodium enters the cell membrane, it generates an action potential, and the way it does that is it repels the potassium, and the potassium wants to get out of the cell. But you're probably wondering, well, how do we reset this? How do we go back into the resting with potential? Because clearly, if I had an action potential that is always being generated, my muscles would be going crazy. Well, yeah, that's true. And the way that it resets itself is that it uses the sodium-potassium pump. So the sodium-potassium pump resets. It resets the cell, the membrane potential. So it resets the membrane potential, and it does this by forcefully shooting out sodium into the extracellular space, and it actively pushes potassium back into the extracellular space. And so whenever the sodium-potassium pump takes potassium from the outside and it pushes it into the cell, we get a really, really large amount of potassium inside the cell. And whenever the potassium inside the cell increases, this becomes a negative number. And so we go from a positive number into a negative number. And so what I'm saying is that this resets the membrane potential. It does not create a membrane potential. Actually, it resets it. So it goes back into equilibrium. So we go from a positive number to a negative number. And that is being done by the sodium-potassium pump. So on an exam, a answer choice will say, oh, the action potential is created by the sodium-potassium pump. And you'll say, no, it's not. It is reset by the sodium-potassium pump, but it is not created by the sodium-potassium pump. Now, within the cell membrane, you have these potassium leak channels. So within the resting, whenever we are resting and we're not generating action potentials, these channels are always open. And so we can say that potassium leak channels are always open. And this allows us to have a constant supply of potassium. So this is a constant supply of potassium. So now let's kind of zoom in and see how this process takes place. Within an action potential, we have about two phases if you're looking at it from kind of like a zoomed out perspective. So here we have a closed section where there is no current, right? So this is going to be in picoamps, which is very, very small current. So whenever we have an action potential, we have a depolarization. So this is a depolarization. And what a depolarization means is that we go into a positive number. So we go into a positive number right there. Now, whenever we need to go from the open to closed, we are going to repolarize. This is repolarization. And repolarization means that we go from a positive number into a negative number. And that is going to be done by the intake of potassium. So sodium leaves the cell, and then potassium enters the cell. And that is going to cause a repolarization. And then we have some time, it would be about 10 seconds over here, where it's not generating an action potential. So this is the closed section, right? So we are not able to generate an action potential. Now notice that this kind of has it has a specific value, right? So within action potential, let's say that we have to hit -50 millivolts to generate an open section. If we hit -49, -48, whatever, we're not going to generate an action potential in this example. And so we have to hit -50. That is the golden number. That is the threshold. And so because we have to hit a threshold, this is called an all or nothing event. So action potentials are all or nothing, meaning that we have to reach the threshold. Okay, so -50 in this example would be the threshold. If I hit -49, -48, that's not really going to do it, right? And you know what? I probably forgot my negative numbers. So -49 is actually greater than -50, so it actually would do the action potential. So what I'm saying is, if you hit -51, -52, something that is lower than this value, then it's not going to work. But if we hit -30 millivolts, then obviously we're going to generate an action potential. Okay, it just has to be greater than this number to generate an action potential. So let's go back to ion channels. So if we talk about ion channels, this is a channel that allows the passage of charged molecules, specifically ions. And we have 3 different channels in this class. In this class, we will be studying the membrane potential ion channels. So whenever a membrane potential shocks the channel into opening, we can allow ions to enter, just for example. So this one kind of uses electricity. So this one uses action potentials. Another ion channel is like ligand-based. So this is a ligand-based ion channel. Okay, and let's say that whenever a little ligand, and a ligand is just a molecule that attaches itself to the channel, whenever the ligand attaches itself into the channel, it actually opens. And when it opens, it allows ions to enter. Now we have mechanical forces. In mechanical forces, you actually use them every day. So a mechanical force, something would physically pull this open. And when this physically pulls it open, it allows ions to enter the cell, right? And this could allow an action potential to occur. And a notable example of a mechanical force used as an ion channel would have to be in the ears. So this would be in the auditory system. So this is the auditory system. And you're probably not believing me, but I will show you how this works. Within your ear, you have those little hairs inside your ear. So if you looked in the mirror and you saw some hairs in your ear, and you'll often see that with older people, you'll see them. And you're probably wondering, well, why do we have those hairs? Well, we have those hairs, and those hairs are called stereocilia. So we have stereocilia. And really, they're attached to each other by these little linkers. So they look like little spider webs, and these little linkers are called tip links. So this is called a tip link. And surprisingly, if you're studying for the MCAT, this will actually show up on the biology section and also the psychological and sociological section. So this is a tip link. And whenever a sound wave enters your ear, it pushes the cilia. And whenever it pushes the cilia, this spider web, the tip link, will pull apart. It will pull apart this mechanical receptor. So this is called a mechanoreceptor. And whenever the mechanoreceptor opens, it actually allows potassium ions to enter the cell. And whenever the potassium ions enter the cell, it causes an action potential. And this action potential generates neural impulses into your brain, and it allows you to hear what's going on. So it gets more complicated, but that's not really the focus of what we're trying to learn. We're just learning that within the auditory system, that there is a mechanical receptor. So this is a good example for a mechanoreceptor. So over here you have the stereocilia, you have the supporting cells, and we have the auditory hair cells right there. And whenever you hear something, it pushes the tip links apart, and it allows for the potassium to flow into the cell, and that generates an action potential, and that can deliver information to your brain. Within the nervous system, we have a bunch of action potentials being generated, and those action potentials can be used to generate neurotransmitters. They generate information. They could pass along different dendrites, right? And so the action potential is going to be inserted over here. And what you just saw is kind of like this wavy form. So why didn't I draw the action potential like a straight line? Well, I drew the action potential like a wavy line because really the action potentials jump, right? So they undergo a saltatorial movement. So they have a jumping movement. So this is a jumping movement. And the reason why they jump is because along this axon, which is kind of like the highway between dendrites, there are gaps within the axon. And these gaps are created by glial cells. So notice how I'm teaching you, and in cell biology, I really shouldn't be talking about this, but I might as well just talk about glial cells and saltatorial movement because it's on the MCAT, and I'm just, passing by it, but let's talk about it. So glial cells are acting like supporting cells within the nervous system. So glial cells actually make the membrane for the axons. And they make the membranes very, very thick. And when these membranes are thick, they are going to have resistance. Okay, so this is a lot of resistance. And if we look at Ohm's law, which is in physics, the voltage, millivolts, is equal to the current multiplied by the resistance. And so if I increase the resistance and I keep this current constant, the voltage will increase, right? And so if the voltage increase, it's more powerful, and it allows us to not waste voltage when it is passing. So when I'm transferring information, the voltage is not going to decrease, it's going to stay relatively constant. So if I have let's say positive 30 millivolts, if I didn't have the glial cells, it would be probably like 15 millivolts right there. But since I have the glial cells, it allows us to stay relatively constant. Maybe it's like 29 millivolts, right? So we don't waste too many volts, right? And what happens here is that the action potential, it cannot leave these parts. But whenever it reaches this section right here, which is exposed, we'll see like the exposed axon, what happens here is that the action potential can jump. So it jumps, right? Let's do this better. It jumps. It can't jump here, it jumps. Can't jump here, it jumps. Can't jump here, it jumps. Can't jump here, but it jumps. So instead of having to travel through here, through here, through here, and through here, that wastes time. And so I'm going to be using these gaps to jump. So that is the jumping effect. And this gap right here, which is exposed, is called a node of Ranvier. So it was created by a French physician. So this is a node, or discovered really. So this is a node of Ranvier, or Ranvier. And so the passage of action potentials go from the axons to the synapse, to the dendrites, and then to the soma. So the soma is kind of like the center portion. It's like this little brown thing inside the dendrite, and that receives the information. So we go axon, synapse, dendrites, and soma, right? So that will be tested on biology exams, MCATs, whatever. So axons, synapse, dendrites, somas. Ax, your sins, dead son, right? Ax, your sins, dead son. That's how I kind of remember. It's very morbid, but you have to get rid of your sins for your dead son. That's how I remember it, but it's pretty cool. So ASDS, right? So here is the action potential, and right here we have a negative 60 millivolts, and the threshold for this system in this example would have to be negative 40 millivolts. So for instance, negative 40 millivolts, that's the minimum that we have to hit to get an action potential. So if I hit, let's say negative 50, I'm not going to have an action potential. If I have negative 45 millivolts, no action potential. But when I hit negative 40 millivolts, then I will have an action potential. So this right here is the threshold value. Okay, so this is the threshold potential. Now the resting membrane potential is actually about, let's say, negative 60. So without any input, naturally this cell membrane would be negative 60 millivolts, and that's the resting potential. But when I hit the threshold, we have a curve, and that curve itself is the action potential. So this right here, when we are increasing, when we're going up, that is the depolarization, polar. And when we go down, that is the repolarization. Now, to create action potentials, we will be abusing voltage-gated sodium channels. So what is a voltage-gated sodium channel? So we're going to call this voltage-gated sodium channel. So what is that? Essentially, without any voltage or without any electrical input, this gate is closed. But whenever we have some sodiums entering the cell, they generate a little bit of electricity. So they become a little bit more positive, until it is so positive that it opens. And whenever it opens, we can actually have more sodiums go into the cell. Okay, so before anything happens, we have little baby amounts of sodium entering the cell, but eventually the cell becomes so positive that it generates an electrical input, and whenever the electrical input is activated, the transporter is going to be opened, or I guess the channel is going to be opened. Now eventually the action potential has to end, and whenever it finishes, the gate closes. Now whenever the gate closes, we cannot immediately create another action potential. There is something called a refractory period, and the refractory period occurs because this inactivating particle, so this is called this little tail right there is called an inactivation particle, activation particle. And this inactivation particle prevents this gate from opening again. So you can have another impulse, right? We can get bit by a dog, and really whenever you get hurt, they send action potentials to the surrounding area, and that generates pain. But whenever the action potential kind of runs out, you cannot have another impulse, right? So my muscles cannot contract for another split second, and they cannot contract because of this inactivation particle. So you cannot have action potentials going like this. Really, it's like this, because this is the refractory period. Okay. And now here we have the membrane potential. So at first we have a closed sodium gate channel. So this is a voltage gated sodium channel, and it is closed. But whenever I hit the threshold, the sodium gates open, and whenever they open, more sodium can enter. And whenever more sodium enters, we have a bunch of sodiums. And when we have too many sodiums, they're gonna take out these little potassiums, they're gonna repel it because they have positive charges, and these sodiums make the potassium leave the cell. And whenever the potassium leaves the cell potential increases, right? So the cell potential increases, and this is called a depolarization. So this is a depolarization, right? Now eventually this has to end, and whenever this ends, the sodium channels become inactivated. So no longer can the sodiums enter the cell. So when sodium tries to, it's going to get blocked. So I cannot enter the cell. But you know what can enter the cell? Potassium, because we're using the potassium pump channels, the sodium potassium pumps, to generate more potassium into the cell. Now, what this picture doesn't show is hyperpolarization. So of course we have this guy right here. So at first we have the depolarization, and this is called a repolarization. Where potassium enters the cell again, but actually the potassium channels, they close slowly. So yeah, we already hit the threshold value. I'm good. I don't need to have more potassiums. But the potassium gate closes very, very slowly. It closes so slowly that sometimes too many potassiums enter the cell. And whenever too many potassiums enter the cell, we have more negative charges. So this is a really negative charge. And eventually these potassium leak channels will allow us to equilibrate. So you remember the potassium leak channels before, where they're always open? Well, they allow the potassiums to go back into equilibrium. And so eventually we go back into a normal position. And so this section right there is called hyperpolarization. So this is hyperpolarization, polarization. And I write really, really badly. So this is hyperpolarization. So we have depolarization, repolarization, and hyperpolarization. So dipolar. So this is a positive MV, this is going into a negative MV, and this is a really negative MV right there. And using this picture, we can see that whenever action potentials are occurring, they only go in one direction. So we can say that action potentials are unidirectional. You might see some textbooks that say propagate, and propagate means to move. So AP, or action potentials, are unidirectional, meaning that they only propagate in one direction. And they only propagate in one direction and not both, so this is incorrect. It would only go to the right side, right? It would only go to the right side because look, we have the inactivated particles. So the inactivation particles, you can't really see it here, but it's blocking the channel, so it prevents the channel from opening. And whenever the channel is prevented from opening, sodium cannot enter the cell. And if we did not have an inactivation particle, then sodium could go that way. The action potential could go to the left, and it can also go to the right. And therefore your muscles would keep contracting, and that you would have a muscle spasm, and that could be kind of lethal within different parts of your body. So thank goodness that we have inactivation particles. So that allows us to move in one direction. So over here we have the action potential being created. So we have the depolarization. It goes into one direction, and eventually these channels will open because sodiums accumulate. They accumulate on this section right here. So the more sodiums we have in this section, they will get near these channels, and the positive charge on the sodium will induce, it will create the channel to be opened, right? So that's why we can actually propagate. So when sodium accumulates on the sides of the cell membrane, it gets near a channel, and the positive charge on the sodium will influence the channel into opening, and that's how we can generate action potentials. So what can we do with action potentials? Well, action potentials can generate signals using neurotransmitters, okay? And so we can use an electrical, we can go from electrical signals into chemical signals, by using action potentials. So the action potential goes from electrical, and it manipulates the synapse into releasing neurotransmitters, and those neurotransmitters can release information. So we go from an electrical signal into a chemical signal, and this is pretty cool. So over here we have these vesicles, and these vesicles, as you can see right here, they contain the neurotransmitters. So this is a postsynaptic membrane, and this is a presynaptic membrane. So this is pre, and this is post. So the action potential is going to generate, it's going to zip along, it will release the neurotransmitters like serotonin, dopamine, whatever, and these little neurotransmitters are going to accumulate between these synapses. And whenever they accumulate between the synapses, they can communicate with the postsynaptic membrane, and they can generate whatever they need to generate. So for instance, if this were acetylcholine, the acetylcholine would go into the muscle cell, which is the postsynaptic membrane, and it would generate muscle contraction. So again, that action potential converts itself into a chemical signal, and in this example, it could generate a muscle contraction. It's very interesting, and we are going to go right into it. So along this presynaptic, so this is going to be pre right here, and this is going to be post right there. And let's say that this is a muscle cell. So because a very common example in biology classes are muscle cells, and so we're gonna see how muscles can contract by using action potentials. So over here we have an action potential, and sodium is constantly going into the presynaptic membrane, and whenever sodium enters the membrane, it generates an action potential. So now we have this guy right here, and it's jumping, and it's jumping, and it's being cute. But look, it reaches a voltage-gated calcium channel. Now before the sodium channel, before the action potential reaches the calcium channel is closed. But whenever the action potential hits the calcium channel, it activates, so it opens. And so now the calcium from outside of the cell can enter, and it reaches the presynaptic vesicle. Now whenever the calcium enters the presynaptic vesicle, the calcium kind of binds to this little vesicle. So the calcium binds to this vesicle, and it's going to eat away at this vesicle. Now, whenever that happens, the neurotransmitter inside the vesicle, let's say that the neurotransmitter is acetylcholine, the acetylcholine inside is going to be released. So now we have little acetylcholines right here, and they're going to diffuse out of the presynaptic vesicle. So now we have little acetylcholines right here floating around in the membrane. Now, eventually, the channel is going to accept one of the neurotransmitters, and so we can say that this is a ligand-based channel. So acetylcholine is going to attach itself to the channel, and it's going to open it. And whenever it opens, the sodium from the surrounding area is going to look at it, and it's going to insert itself. And whenever the sodium inserts itself into the membrane, it generates action potentials. And whenever this action potential is inside the muscle, in this example, it generates a muscle contraction, which is so amazing. And so what I'm trying to say is that voltage-gated calcium channels, so voltage-gated calcium channels are used to convert electrical signals into chemical signals. They convert electrical signals into chemical signals. And you know what? That is so amazing. Like, isn't that exciting? Like, are you not hyped? It's just, I don't know. It's so like spectacular. I'm absolutely speechless. And I just stuttered. So if you hadn't noticed, I do stutter. So that's pretty cool. So yeah, it's just, I don't know. I get goosebumps talking about it, but it's just sodium enters, it generates an action potential. The voltage-gated calcium channel opens, and it generates this acetylcholine. Or it doesn't generate, but it releases the neurotransmitter, and the neurotransmitter goes into the ligand-gated channel, and it's going to generate another action potential. So let's kind of look at it in more detail. And so here is a zoomed up picture of the ligand-gated channel. So some people, they call that a ligand-gated channel, and some people, they call it a transmitter-gated channel. Whichever you like, you can use it. But here we have, in this example, the acetylcholine binding sites. So we would have the presynaptic vesicle right here, and it releases the acetylcholine. So we have the acetylcholine kind of releasing. This is a vesicle, by the way. And let's kind of do a different color. So again, we have the acetylcholine right here, and the calcium is eating away. It's not really eating, it's just kind of allowing reactions to take place. But the calcium allows the vesicle to kind of fuse with the membrane, and whenever it fuses with the membrane, the acetylcholines will be released into the environment. Right? And so it will go into the acetylcholine binding sites. So whenever acetylcholine goes to the binding sites, it is going to allow this channel to open. And whenever this channel opens, the sodium goes inside. And whenever the sodium goes inside, it goes right to the cell membrane, and we start to have an accumulation of sodium. And whenever we have an accumulation of sodium, the other channels, let's say that we have a sodium channel. So we have a sodium channel right there. The sodium channel will actually depolarize, and it will allow more sodiums to enter. And so we have another propagation of an action potential. Right? So that is how action potentials can accumulate. And these action potentials can go and they can stimulate muscle, and that's how a muscle contraction is activated. So specifically for muscles, you will use acetylcholine. So you won't have muscle contraction for dopamine or serotonin, but really you will use acetylcholine to work with the muscle. But you're probably wondering, okay, well, if this happens and we have an accumulation of acetylcholine, how do we not have muscle spasms every day? How do we get rid of that? Well, there are two ways. Well, really, there are three ways that we can get rid of this acetylcholine, or the neurotransmitter. So for instance, let's say that over time the acetylcholine kind of diffuses away. So what I'm saying is, over time the acetylcholine just kind of drifts away and it goes somewhere else. And so there is no more acetylcholine in this section. Another way that we can get rid of acetylcholine is to get an esterase. So we can have acetylcholine, we can have acetylcholine esterase. And what this enzyme does is that it will bind itself to the acetylcholine and it will destroy it. Right? So now we just have little pieces of acetylcholine. And these little pieces of acetylcholine go back into the presynaptic cell, and they are recycled. And so now we will have more acetylcholine to be used next time. Now another way that we can prevent muscle contractions is by physically blocking the active sites. So if we block the binding sites with something else, then the acetylcholine cannot bind to the binding site because it's already taken up. And so that can get rid of any muscle contractions. And so toxins, they really like to do this, right? So they really like to block the binding sites. Now some toxins can be permanent, and so now you can never have acetylcholine binding to this binding site, and so that could lead to permanent muscle atrophy, muscle weakening. You can't move your arm anymore because all these binding sites are blocked. And so that's how snake venom most often works. Sometimes you have Botox. So Botox is kind of like a little bit of toxin, and that can block the binding sites. And what happens then is that your wrinkles, they kind of smooth out because now your muscles cannot contract. Wrinkles are muscle contractions, and whenever the muscles cannot contract, your wrinkles go away and you appear to seem younger, right? In the case of drugs, for instance, medications for people who have mental health issues like depression, sometimes they're not producing enough serotonin. And the serotonin that they do produce is whisked away. It goes away, or it diffuses away, or it's taken up by the vesicle, right? So it can be taken up by the vesicle. But what if I modified something? What if I prevented the serotonin from being recycled, or prevented the enzyme that breaks it down? Well, that means that I would have more neurotransmitter within the synaptic cleft. So this whole region of space is called a cleft. C-L-E-F-T, cleft. And so if I prevent the neurotransmitter from being broken down or diffusing away or being taken up by the presynaptic vesicle, then there is more neurotransmitter that is able to bind to the binding sites. And that could allow us to feel happiness, right? Which is pretty cool. Whenever you snort cocaine, and I hope that you never do, these neurotransmitters are being activated. So the cocaine allows us to have a constant supply of serotonin, right, and dopamine. So whenever that happens, you feel happy and you want to do it, right? So your serotonin levels are really, really up. So whenever you snort cocaine, the serotonin goes up and the dopamine goes down. So the dopamine, it kind of acts as a controller. So if we have high levels of dopamine, it kind of says, okay, we're done. We don't need to eat any more cake. I'm full. I'm happy, right? So that levels satiation. So that makes us feel content. It makes us feel full, right? But whenever we snort cocaine, the serotonins go up and the dopamine goes down. And so now we don't have a controller that says to stop. We just want to keep doing it, and we keep doing it so much. Eventually, these channels, they become used so much that they stop responding to stimulus. And whenever they stop responding to stimulus, the normal neurotransmitters cannot create an action potential. And whenever they cannot create an action potential, they don't respond to it, and we don't feel happy anymore. So that's why when people do coke a lot, in this example, you often see them feel depressed and sad, and they have to do more and more amounts of cocaine until they eventually overdose and die. And that is because the membranes become so used to having a lot of neurotransmitters at one time. And so that's why cocaine addictions are so deadly, and they kill a lot of Americans, most likely from the higher end of the social class and the economical class. Right, so serotonin goes up, dopamine goes down whenever you're snorting cocaine. So hopefully now you know not to have a cocaine addiction, because it can kill you, and it's also very, very expensive. And before we move on to the next slide, I have to mention that we have two types of neurotransmitters. We can have excitatory neurotransmitters, and we can also have negative neurotransmitters. So this is inhibitory, inhib, and this is exciting. So an inhibitory neurotransmitter would have to be like a sedative, right? And this could be GABA, which stands for, I don't know, some nerd stuff, right? But essentially, this is a negative neurotransmitter. An exciting one would have to be acetylcholine, right? But usually, these right here, these are cation channels. So these are cation channels, which are exciting. And the negative channels would have to be chlorine, right? So we have chloride channels. And that's kind of like the general rule. So if you have an exam and it says what ion is typically used for inhibitory channels, you would say, oh, we're going to be using chloride channels because they're negative. What ion is used for exciting channels? Then we use sodium or potassium, right? Because those are cations. Those are positively charged ions. So scientists are kind of messed up if you think about it. All the things that we ever made have been tested on animals, and neurotransmitters and action potentials are not the exception. They have to be tested on animals. So this dude right here, he's not having a fun time. He has a wire connected directly to his brain, and this wire is constantly flashing a blue light. If we turn off the light, we have a different reaction to the rat. So let's say that the rat is normally calm, he's chill, but whenever we turn on the light, action potentials are going to be generated by this light, and sometimes, depending on where you place it, the rat will become aggressive. And so here on this picture, when the light is off, the rat is chill. You know, he's probably thinking about making food, like his cousin from that Pixar movie. But whenever the light is turned on, the rat will actually fight this glove. So there's a glove right there. So whenever the light is on, the rat becomes aggressive and it starts trying to kill the glove. But when the light is off, then the rat is chill, he's calm, and he says sorry. And so this right here is called optogenetics. This is called optogenetics. And scientists are currently trying to figure out how light, with this frequencies, how it can stimulate action potentials and how it can release neurotransmitters into different parts of the brain. We don't know which part to put it in to relieve depression, to get rid of possible blindness, to get rid of muscle contractions, or sometimes people can't move their arms. And so maybe this could be a cure to muscle inactivation, right? So whenever you shine light on different parts of the region, that could make the arm move. So we're trying to figure out where to put it in these rats. And someday they may put it on people. You may never know. And the way they use it is that they use channel rhodopsin. So again, channel rhodopsin is a transmembrane channel, right? And they put that into the brain of the rat. And then they have the fiber optic cable, and essentially it uses this blue light, and blue light has a lot of energy in it. Now the channel rhodopsin is going to use the energy from the blue light and is going to open the channels. So here's the channel rhodopsin right here, and it uses the light from the blue light to open, and when it opens, it allows sodium to enter the cell. And whenever the sodium enters the cell, it generates an action potential that could release neurotransmitters later on down the line, right? So whenever these sodiums enter the cell membrane, they could release neurotransmitters. It could cure depression by releasing serotonin. It could cure muscle inactivation by releasing acetylcholine. It could release dopamine. We don't know. It could even cure blindness, but that is a rare case. So hopefully now you understand active potentials, transmembrane channels, active diffusion, sorry, not active diffusion, but active transport, passive transport, osmosis, and all that stuff. So I hope that you have a great day and that you had fun learning about all these cool things. So this is cell biology, and I hope that you have a great day. Remember that you are amazing and that I absolutely love you. Okay? So have a great day and be safe. Thank you.